Liver diseases. Hemochromatosis is a well-defined syndrome characterized by normal iron-driven erythropoiesis and the toxic accumulation of iron in parenchymal cells of liver, heart, and endocrine glands. It can be caused by mutations that affect any of the proteins that limit the entry of the iron in the blood. Hereditary hemochromatosis is caused by a mutation in a gene that controls the amount of iron in your body absorption from the food you eat. This type is by far the most common type. The initial symptoms of hemochromatosis can include feeling very tired all the time, weight loss, weakness, joint pain, irregular periods or absence of periods, and inability to get or maintain an erection. The pathogenesis of this disease that if one or more components of the blood iron sensing machinery fail, adequate levels of hepcidin will not be produced in response to the increased levels of iron. Intestinal and macrophage iron release will not be checked, and iron overload and hemochromatosis will result. Hepcidin deficiency has been associated with HFE associated, HGV associated, and TFR2 associated and HAMP associated hemochromatosis. The management of this disease is by bloodletting. Phlebotomy is the standard treatment for all forms of hemochromatosis. The treatment is conventionally initiated when serum ferritin levels exceed the normal range. Cirrhosis is due to progressive iron deposition in the liver parenchyma, and it is one of the most common disease manifestations of the tissue damage caused by hemochromatosis. The Wilson disease is a rare genetic disorder characterized by excess copper stored in various body tissues, particularly the liver, brain, and corneas of the eyes. The causes of this disease is by a mutation in the Wilson disease protein ATP7B gene. This condition is autosomal recessive. For a person to be affected, they must inherit a mutated copy of the gene from both parents. Now, the pathogenesis of this disease is that in response to the risen concentrations of copper, ATP7A, Menkes protein, releases copper into the portal vein to the liver. Liver cells also carry the CMT1 protein and metallothionein and ATOX1 bind it inside the cell. But here it is ATP7B that links copper to ceruloplasmin and releases it into the bloodstream, as well as removing excess copper by secreting it into bile. When the amount of copper in the liver overwhelms because it is untreated, Wilson disease can be fatal. The proteins that normally bind it causes oxidative damage through a process known as Fenton chemistry. This damage eventually leads to chronic active hepatitis, fibrosis, and cirrhosis.